Hi, it's Miss Bastido, and today we're going to be talking about Unit 4, Topic 1, and specifically modes of inheritance and how different traits are inherited. This is a pretty simplistic view of how traits can be inherited, but good for a foundation in genetics. So this all really, some historical background is that Gregor Mendel, known as the father of genetics, was working in a garden and he was studying pea plants, and he was noticing different traits in pea plants and how they were inherited. And so what he's looking at here, he took two pea plants. He called these the F0, or sometimes we call it the pea generation. And he found some pea plants had purple flowers and some had white flowers. He took some that, normally pea plants will self-pollinate. And so that self-pollination produces, they're still producing sex cells, but they're self-pollinating. He took purple flowers that had come from other pea plants with purple flowers, and so he knew for sure. We call them true breeding. That means that they would only produce more purple flowers. And then he crossed that with a white flowered pea plant. So because pea plants were self-pollinating, he actually had to sort of mechanically do this and cut off the male parts of one flower and dust the pollen from one flower onto the other. But what he found was in this F1 generation that all of the flowers were purple. And so he was wondering, where did the white flowers go? And so at the F1 generation, he got 100% purple flowered. Then he just let this F1 generation self-pollinate as they normally would. And what he found was they ended up with a 3-to-1 ratio of purple to white flowers. And so this white flower that had somehow disappeared in the F1 generation reappears in the F2 generation, albeit in a smaller proportion than it had been in the original F0 generation. And so what he found from this was that all traits come with different forms. And so we call these forms, so the purple and the white flowers, we call those alleles. And so these alleles are found on chromosomes, and the chromosomes are what we inherit. Of course, Mendel didn't know all of that, but you have two alleles for every gene, one from your mom and one from your dad. And what that literally means is you have two chromosomes, one from your mom and one from your dad, and that's where your alleles are located, or your genes are located. And so the difference in the nucleotide sequence in this spot in the DNA for this gene determines what allele you'll end up with. And so just slight differences in A's, T's, C's, and G's in that gene can cause you to have, in this case, a purple flower or a white flower. And so we'd say those are just two DNA variations for the gene for flower color. And these are located on those homologous chromosomes, which we talked about in the meiosis video. So what he found was that some traits are masking others. And so if you're carrying an allele of each flower color, that somehow we didn't end up with a blend, but instead we ended up with one dominating over the other. And so purple plus white didn't give us like a lilac or light purple color. Instead, what it gives us is all purple. And this really means that the dominant allele is the functional protein, and the recessive allele is not functioning or not being expressed to the same extent as the dominant allele. The recessive allele obviously makes a protein that's either malfunctioning, although more likely that it's just being masked by the other one. And so having that purple allele makes the white allele not really be seen. So remembering that the wild type, we sometimes refer to it as wild type, although you don't have to know that, is that that's the allele producing the functional protein, and then the mutant is the one producing the protein that's not functioning properly. So when we look at this, we can look at the genotypes and the phenotypes. So these are some terms that you'll need to know. A phenotype is the description of the physical characteristics of an organism. And so when we look at the purple and white flowers, the phenotypes are purple or white. The genotype is their genetic makeup. So what do those alleles show? What are their alleles that they have? And so if we were explaining these results, I could say a purple flowered, the phenotypes for the P or the F0 generation here are purple and white flowers. Those are the phenotypes. The genotypes here, because they were pure breeding, the genotype for purple would be 2 purple alleles, whereas the genotype for white would be 2 white alleles, which we do usually use letters to denote that. And then in the F1 generation, we end up with all purple phenotype, but the genotype is that they're carrying both a purple and a white allele. And so we can show this in a Punnett square. And so this is just a way to predict probability when you're doing a cross. And so here's my F1 generation with 2 purple flowered plants. That's the phenotype. The genotype is that they're both carriers of the recessive allele. So they are heterozygous. And we would put the one parent on one side and one parent on the other side. And then we just fill in the boxes. And you can see that we end up with this purple flower here. We have a purple flower here. This one will also be purple, and this one will be white. In doing that, we end up with this ratio of 25% in our genotype being having 2 purple alleles, 50% having a purple and a white allele, and 25% having 2 white alleles. But if I put the phenotype here, the phenotype is what we see. We actually have 75% that are purple and 25% that are white, which gives us that 3-to-1 ratio. And so we would say, in using the terms that I said earlier, homozygous means that there's 2 of the same alleles. So all recessive, if an individual is expressing the recessive phenotype, they will always be homozygous. But for the dominant phenotype, they could be homozygous, meaning they have 2 of the same alleles, or they might be heterozygous, meaning they have 2 different alleles. And so to be purple, you could be, to have purple flowers in a pea plant, you could be homozygous for purple, or you could be heterozygous for purple. And so we could be homozygous dominant, homozygous recessive, although just saying they're recessive is implying that they are also homozygous. And so while what Mendel found was significant, there's a lot of traits that are not inherited so simply. So Mendel actually found 7 different traits in pea plants that were all inherited that way. That mostly worked because those genes were found on different chromosomes, but also not all traits are inherited so simply. And so we're going to go through just a few different examples of other modes of inheritance. And so the first one is incomplete dominance. And so in some individuals, we have, these are carnations actually, but also this is true of Japanese 4 o'clock flowers. And so in this case, we have red flowers and we have white flowers, but the intermediate or the heterozygote gives us a pink flower. And so that's called incomplete dominance when there's a blend between the 2 alleles. Really, the better way to write these are with 2 different letters, because using a lowercase letter implies that one is recessive and one really is not recessive. They are incompletely dominant. And so you have, to be red, you would have 2 red alleles. To be white, you have 2 white alleles. And to be pink, you have a red and a white allele. So here's some Japanese 4 o'clocks. If we had true breeding red flowers and true breeding white flowers, so this would be big R, this is WW, then we have 100% pink flowers in the F1. If you were to cross that and let those self-pollinate, we end up with red flowers, 50% pink, 25% white. And so that's a bit different than what we found in the dominant recessive where we had a 3-to-1 ratio, because now that 50% that would have been red before, would have been purple before in our other example, now it has its own color of pink. Another example is codominance. And so in codominance, both alleles are expressed not in a blended form, but they're just both expressed. So the best example of this is human blood type. And human blood type is an example of a dominant recessive trait. It's an example of a codominant trait, but it's also an example of a multiple allele trait that means that we have more than 2 alleles that control the gene. And so in blood type, we have an A allele, a B allele, and an O allele. Notice that we write these a bit differently, and this is just the standard way to write it. So A and B are co-dominant with each other. But A and B are both dominant to O, and we use a lowercase I for the O. And all these represent the A's, the B's, is antigens, which are proteins found on the surface of the red blood cell. And so if you have type A blood, that means you have an A antigen on the surface of your red blood cell. If you have type B blood, you have the B antigen on top of the surface of the red blood cell. And if you're type AB blood, then you have both of those produced. If you have type O, or if you have a lowercase I allele, that one is recessive to both A and B. So you could have type A blood and be heterozygous. You can have type B blood and be heterozygous. You could not have type O blood and be heterozygous. So here's what this looks like. If you have type A blood, then you can either be this or this. Those are your 2 possible genotypes. You have the A antigen on the surface of your red blood cell. I'm not going to talk too much about this, but that means that you produce an antibody against the B antigen. And that's mostly because you consider it foreign. Your body considers that a foreign antigen. And so if you were to give somebody with type A blood, type B blood transfusion, their blood would coagulate or clump because the antibodies would start attacking that blood. If you have type B blood, you can be B, or you could be B, or you could be BO. And that means you're still producing the B antigen on the surface of the red blood cell, but you produce an anti-A antibody, which means you can't give people with type B blood type A transfusions. If you have type AB blood, you produce both. And this is the only possible genotype because these are co-dominant with each other, which means you have some A antigens and some B antigens. You don't produce any antibodies against these antigens because you consider them both self, because you have both of them. This means because you're not producing any antibodies against any blood type, you are the universal recipient. If you have type AB blood, you can receive any kind of blood. In type O blood, you're recessive. And so you have 2 recessives. You have no antigens on the surface of your red blood cell, which means you're producing antibodies against both A and B antigens. That causes you to only be able to receive type O blood, but you could donate to anyone. You're probably thinking, how does the positive negative fit into this? That's a separate gene, and that gene is a dominant recessive gene, with positive being dominant to negative. People with positive blood can receive positive or negative blood. People with negative blood can only receive negative blood. You don't need to know a lot of details on that. A bit of an interruption there, but you don't need to know a lot of details about the antibodies and the antigens. The key is that you can do Punnett squares using these alleles and understanding that A and B are co-dominant and that they are both dominant to O. Another mode of inheritance is polygenic inheritance. And so this is when it gets a bit more complicating, and doing a simple Punnett square by itself is not going to suffice. And so polygenic inheritance, and the prefix poly means many, and then genic talking about many genes. So many genes are controlling the trait. And we usually say if 2 or more genes are controlling the trait, then we'd say it's polygenic. And so you can think of anything where the phenotype is on a continuum. And so there's lots and lots of different options, not just 2 or 3. And so lots of human traits are polygenic. One could argue most, but things like skin color and height and weight, obviously some of these have also environmental factors that affect them, but all of these would be on a continuum and be polygenic. So you should be able to predict, though, with up to 3 genes how this would be inherited. And so if they've given you the genotype of a parent, so you can look at this one here, this is of 3 different genes. Both parents have, in this case, a medium skin color, and they are heterozygous for each of the 3 genes. You could do one giant Punnett square to figure this out, or you can just do 3 different ones. So I would just do a Punnett square with big A little a and big A little a, and then do another Punnett square with big B little b, and then another one with big C, big C, little c. And then when I ask you what are the probabilities that you end up with somebody little a, little b, little b, little b, c, little c, then you would just multiply the probabilities. So if I did that Punnett square, which I don't have it here, but I would end up in the first case, you just have 1/4 of a chance of getting little a, little a. In the second chance of the second Punnett square, you'd have 1/4 of a chance getting little b, little b. And then in the third one, you'd have 1/4 of a chance getting little c, little c. Take those 3 1/4s that you got, and you would multiply those probabilities together. So 1/4 times 1/4 times 1/4 gives you 1/64, and that's where this comes from. So it is possible to do a really huge Punnett square with 3 genes, but sometimes we can really make mistakes when we're trying to fill those in. So I really recommend doing 3 separate just normal squares and then multiplying the probabilities that you get whatever they're asking for on an exam. And we'll do some practice problems of that in class. The last mode of inheritance is sex-linked traits. And so these are genes found on the sex chromosomes as opposed to all the other chromosomes. So either on the X or on the Y, although typically we just refer mostly to sex-linked traits. They're mostly on the X chromosome. This was discovered by Thomas Hunt Morgan when he was breeding fruit flies. And so fruit flies are really common genetic subjects because they have a really fast breeding time. They're called Drosophila melanogaster, and they have 2-week generations. They have 4 pairs of chromosomes. They're XX female and XY male, similar to humans. And so they've lots of genetics courses that you might take in uni you would do some work with fruit flies and to learn this sort of thing. But remember that in humans we have 22 pairs of autosomes, and we have 2 pairs of sex chromosomes, or sorry, 1 pair of sex chromosomes, either an X and a Y or an X and an X. So those are the sex chromosomes. So when he discovered this, he had true breeding red-eyed females. And I gave you a little buzzing sound there for you. So we have red-eyed females, and he had white-eyed males, and he let them cross-breed with each other. And in this F1 generation, he ends up with everyone having red eyes, which you would expect if it was a dominant recessive trait. So both the males and the females all end up with red eyes. But then he lets the F1 generation, he allows them to mate with each other, and he ends up with all of the females having red eyes, and only 50% of the males having red eyes, and 50% having white eyes. And so he said that it must have something to do with their sex, because why are only the males ending up with white eyes? And so we know there's 2 sex chromosomes. You either have 2 X's, develop as a female, and are assigned female at birth. And that is a bit of redundancy, meaning you really only need 1 X, because obviously males live fine with just 1 X. And so there's no redundancy there. There's a Y chromosome. So when we have sex-linked traits, we would use the XY. They would have to tell you on an exam that these were sex-linked traits. There's no expectation that you know every sex-linked trait that's out there. And so you can see that there's always this 50-50 chance of having male or female assigned at birth, depending on which chromosome is inherited. So if we're looking at his fruit flies again, we can do a Punnett square with this. And so here's what happened. He had red-eyed females. They must have been homozygous for red eyes. Remember, this is on the X chromosome. And he had white-eyed males. And so if we put that into the Punnett square, we end up with these 4 genotypes. And everyone ends up with red eyes. Notice that there's nothing written on the Y chromosome because the gene is not found on the Y, it is only found on the X. If we allow these guys here to mate with each other, then we would have this scenario. So now we have females that are carrying the white-eyed gene, and males that are red. And so if we fill that Punnett square in, you can see here that we end up with, this would be a homozygous red-eyed fruit fly. This would be a a red-eyed male. This would be a heterozygous red-eyed female, and then we would end up with a small chance of having a white-eyed male. And that the key here is that he's really inheriting the white eyes from the mother, from the X chromosome from the mom. There are lots of genes linked to the human X chromosome. There's a bunch of them listed here. And usually when we say something is sex-linked, we're usually really referring to it being X-linked. Lots of diseases are traced on the X chromosome, but also just remembering that males are more likely to get this trait because they only have one X. So if they get the recessive, they have it. They don't need to have two recessives, whereas females would have to receive two recessives to get a sex-linked trait. I hope this video was useful to you. I know it was a bit quick, but hopefully with some extra practice, recommend really working on just practice problems for Punnett squares and inheritance, as that's the best way to be able to do these problems on an exam. Let me know if you have any questions.